So in the first lecture, I stressed the importance of protein phosphorylation for biology. And in the second lecture, I tried to articulate for you the protein kinase molecule, the three-dimensional molecule, and how it works. What I'd like to do here is to focus on how that kinase is regulated and how it is localized. And so again, I'm going to focus on pKa as the model system, in part because it's the kinase we really understand the most about in biochemical terms and structural terms. So again, it is activated by a hormone binding to a GPCR, which then leads to the generation of cyclic AMP, and then that unleashes the catalytic activity of pKa, the kinase. What I'm going to focus on today is this other side where it is in its inactive state and also targeted to specific sites in the cell by a kinase anchoring proteins. And this is really an essential feature of specificity and biological function for pKa. And so we're going to look at the holoenzymes. I showed you previously what we learned from the structure of the catalytic subunit by itself. What I'm going to tell you here is how much more information we obtained from having a structure of the holoenzyme complex. And then I'm going to focus on the A kinase anchoring proteins, tell you how they target pKa to specific sites, and again really leave you with the global challenge for all of us to understand how these kinases exist as part of larger, often very large macromolecular complexes. So if we look at that kinase that I told you about before, we have the structure of pKa bound to an inhibitor peptide, and I've told you about the active site residues. And what I want to look at now, here's the kinase with the two lobes with ATP bound at the cleft between the two lobes. And I've told you about those subdomains. I think you have a good concept of those subdomains now. What we're going to focus on now is the surface of that kinase, because it's the surface that the kinase uses to interact with other proteins. And these kinases are protein interaction modules. They communicate with other proteins, and they use their surface to do that. So here's the pKa with its conserved subdomains, and now if we look at that core of pKa, you can see its crevices, the charge features of that surface. Each kinase is quite different on its surface, and that dictates what proteins it will interact with. So the major protein, one of the major proteins that binds to the catalytic subunit are the regulatory subunits, and it's multifunctional, modular, and highly dynamic. It has two cyclic AMP binding domains at the C-terminus, and then it has an inhibitor sequence that binds to the active site cleft of the catalytic subunit, and then at the N-terminus it has a dimerization domain. This linker region, in the absence of cyclic AMP or catalytic subunit, is a very dynamic entity, this linker region. And so it's hard to do structure of the whole thing, but we could do structures of the monomeric forms, and they're perfectly functional in terms of binding cyclic AMP and binding catalytic subunit with very high affinity, sub-nanomolar affinity. And so we did structures of this one. We've also done a structure of the monomeric form. This first structure in 1995 showed us the cyclic AMP binding domain and told us how this regulatory subunit functioned as a receptor for cyclic AMP. And cyclic AMP is a major signaling molecule in all of biology. It's a second messenger signal of external stress in bacteria as well as in man. And the domain that it binds to, this cyclic nucleotide binding domain, is also conserved. So it's binding to this domain that now translates that cyclic AMP signal into a biological response. And so the general features of this domain were really elucidated by the structure of the cyclic AMP-bound R subunit. And you can see the same domain is conserved in a number of other proteins. So looking more closely at that motif, there is what we call a phosphate binding cassette, this PBC, which anchors the phosphate of cyclic AMP, another very important phosphate in biology. And then in addition, you have part of that conserved motif are these B and C helices, which are functionally conserved in all of those domains. And each cyclic nucleotide binding domain has a hydrophobic capping residue, so you can see that the adenine ring is capped between two hydrophobic residues, and this can come from different parts of the molecule, but the PBC, the B and C helices, these are conserved. And so if we now look at what we have learned from the individual proteins, from the cyclic AMP bound structure of the regulatory subunit and from the free catalytic subunit, we've learned a lot. How does this function as a receptor? How do we understand that cyclic AMP binding motif? How does it work as a catalyst? So we've learned a lot. But we didn't really understand how is the catalytic subunit actually inhibited by the regulatory subunit, nor did we understand how the holoenzyme is activated by cyclic AMP. And these were things that we really have learned just in the last few years. And for me, it has certainly demonstrated to me the importance of having larger complex to be able to really understand function. And certainly we have understood so much more about the function of pKa having a structure of a holoenzyme complex compared to what we knew with just the free catalytic subunits and regulatory subunits. So this is the small single cyclic AMP binding domain protein bound to the catalytic subunit, and you can see several things from this structure. First, the red part is the inhibitor peptide, which was always disordered before. Now it becomes ordered at the active site cleft. The major interactions here of this surface are with the large lobe. So the large lobe is an important docking site for many proteins to bind to protein kinases. Okay, so let's look more closely at what was really surprising to us about this structure. We really didn't anticipate this at all. So on the left, we have the structure of the cyclic AMP bound domain. And then on the right, I'm going to show you what happens when you release cyclic AMP and you bind to the catalytic subunit. And you can see that a major conformational change, the helical subdomain changes, and this inhibitor peptide becomes ordered. That's ordered at the active site cleft. And in this structure, that inhibitor region was there, but it was always disordered. I told you that linker is very disordered when it's a free regulatory subunit. So let's look at first this ordering of the inhibitor site. So I told you in the last lecture how pKi, a small inhibitor peptide, is bound to the active site cleft. And they have a common mechanism. This region that's circled here is the common inhibitor region. It also mimics the substrate. So that binds directly to the active site cleft. And they do it in the same way. pKi, on the other hand, bound with that amphipathic helix, that achieved, again, subnanomolar affinity, but on a different surface. Whereas here you can see the regulatory subunit is using a completely different surface to achieve high affinity. And I'm going to show you this again so you can appreciate the dynamic features, especially this BC helix, how that BC helix goes from a kinked structure to an extended structure. So a very, very dynamic module, and we didn't anticipate such a conformational change at all before seeing the structure. So here you can see, again, now we've docked the inhibitor peptide, and then you can see as you morph from the cyclic AMP bound conformation into the bound conformation, you can see that C helix, how as it extends, it binds onto the surface of the large lobe. So it makes a major contact with the large lobe. So this is the activation loop that I told you about before. Here's that phosphate. This is an active kinase. The regulatory subunit doesn't bind very well if it's not an active and phosphorylated kinase. And so I also told you that this threonine feeds back to all those different parts of the molecule, including the active site, makes an active kinase. So if we look where that helix from the regulatory subunit is docking, it's docking right up against that activation loop. And this is a critical interface. So we now have a further understanding of what you achieve by phosphorylating this kinase. You create an active kinase, so you stabilize the active site so it's optimized for catalysis. But the other thing that you do, based on the holoenzyme structure, is that you also create a surface here that is now providing a docking site for the regulatory subunit. So again, it impresses, I think, on you and all of us the importance of one single phosphate on the functioning of this protein. This is now both domains, and the question was, does that helix extend in both domains? And of course it does. It's even longer. And so the consequences of this are really very important in looking at this entire structure. Again, it's a very large interface that you have here for the A domain, the B domain binding to the catalytic subunit, and looking at it as a static difference. This is the cyclic AMP bound state where this inhibitor site is present but disordered. You don't see it. And then now when you look at releasing cyclic AMP and binding to the catalytic subunit, you now splay apart the two domains. You have a very long extended helix that separates the two domains, and you have the inhibitor peptide docked to the active site cleft. So it's a major conformational change that you induce in the regulatory subunit as a consequence of releasing cyclic AMP and binding to the catalytic subunit. And the catalytic subunit does not change very much, its conformation. And so this is just letting you see this again, flipping back and forth, the change in the proximity, the organization of these two domains. And so you can look, just going back and forth, how that B domain is really just hooked onto that helix, and so when that helix extends, it flips away the B domain and really dissociates the two domains from one another. Okay, so we've looked at the consequences of binding the regulatory subunit, how the C subunit is inhibited, how it gets activated by cyclic AMP. That came from having a holoenzyme structure. Let's look at this other feature. The kinases, pKa is not typically just floating around in solution. It's often targeted to specific sites. And if you want to look at the dynamics of time and space as part of your signaling equation, then you have to look at how is pKa localized. And in large part, this is achieved by A kinase anchoring proteins, scaffold proteins that bind with high affinity to the regulatory subunit. So let me explain that a little bit more. These are just some examples that John Scott gave me of different places in which the regulatory subunits can be targeted, to the plasma membrane, to mitochondria, to all different sites, so that you have localized communities of signaling. And if we looked at what I've told you so far, is this part of the molecule that's important for binding to the catalytic subunit and inhibiting the catalytic activity. It's this part, the dimerization domain, that is important for targeting to AKAPs. And we've looked in particular at dual specific AKAPs. Most AKAPs bind to the R2 subunits. They bind with very high affinity in the nanomolar range to R2 subunits. There are some that bind to both R1 and R2 subunits, and those we call dual specific AKAPs or DAKAPs. So just to again give you a sense of the complexity of this signaling system, this is the L-type calcium channel where you have one of its tails regulated by a pKa phosphorylation site. We have this extraordinary advances in getting structures of channels and membrane proteins and understanding for the first time at atomic level resolution how an ion passes through a channel. But it's often the tails and the loops that are disordered on the cytoplasmic side that provide the mechanism for regulating whether that channel is open or not. And this is the case for this L-type calcium channel. There's a pKa phosphorylation site there. And pKa is targeted to this site by this AKAP 179, which has many binding partners, but it targets to pKa by this little mechanism. It's just an amphipathic helix in the AKAP. And so you bring the kinase there. You have PP2A as a phosphatase, calcineurin as a phosphatase. Very often you have in this system kinases and phosphatases that work together in a synergistic way. And it's this synergy, which is another thing that I hope to convey to you, how these molecules work together, how different parts of one molecule, like the kinase, work together to create an active enzyme, and how different molecules come together to create a signaling unit. So one example, DACAP1, targets pKa to the outer mitochondrial membrane. And if we look at DACAP1, these AKAPs have many binding modules, and DACAP1 has a little pKa binding motif there that I'll explain to you further. And it also has a targeting. That's what takes it to the mitochondria, the N-terminus, and if you add an extra 30 residues at the N-terminus, it goes to ER. So DACAP1 is an example of one of these AKAPs. And here you can just see, if you ordinarily R1 in these cells is cytoplasmic, but if you co-express R1, which is labeled with the GFP in green, with DACAP1, you find that it all goes to the mitochondria. And this is very characteristic of the mitochondrial morphology in the cell. Okay, DACAP2 is another one that we've looked at, and DACAP2 binds both to R1 and R2, and it regulates ion transporters. And so this is another big function of these AKAPs, to regulate ion transporters. And so the ion transporter that was first shown to regulate is the sodium phosphate ion transporter, and this is regulated by, not by phosphorylation, but by internalization. When you don't want, this is in kidney proximal tubules, when you don't want phosphate to be taken up, you internalize the transporter. And DACAP2 is linked to the transporter through a PDZ domain protein, and these are little binding modules, PDZ domains, that now link the AKAP to the cotransporter and pKa through the AKAP. So let's look more closely at DACAP2 and just its binding motif. So the C-terminus of DACAP2 contains all the information that's important for binding to pKa. It's this little region in red here. It's modeled to be an amphipathic helix. It is an amphipathic helix. And then this little bit at the end, the 3 residues at the end are what target it to that PDZ domain. But it's going to be this amphipathic helix that we took this peptide and crystallized it together with the dimerization domain of R2 alpha. And you can see here this complex, the red is the AKAP peptide, binds with 2 nanomolar affinity very, very tightly. And you can see the N-terminus is a 4-helix bundle of both chains of the regulatory subunit. And then most recently we did the structure of the R1 dimerization domain. Here, where this has a lower affinity, 50 nanomolar, but critical for docking, particularly under stress conditions of targeting pKa, the R1 subunit. So the R1 subunit is typically more soluble than the R2 subunits. Our R2 subunits are mostly targeted. Our R1 subunits are cytoplasmic, but they can be dynamically recruited. If you cap a T cell, for example, R1 is normally soluble. You cap it with an antigen, R1 goes to the cap site. So R1 is much more dynamic. And it also has disulfide bonds that actually link the 2 protomers. So we have a chemical understanding of these docking motifs. And here is just showing you, again, that R1 subunit, how this amphipathic helix is binding specifically to the surface, and seeing that as a surface, a very close packing of the small helix to this dimerization domain. And when you remove the peptide, you can see this groove. It's a very characteristic groove and quite different for R1 and R2. So that's how you achieve targeting. So let's look at one more feature. I told you how important it was for us to be able to understand the holoenzyme complex, a complex of a monomeric R subunit and C subunit. So in the physiological state, R1 exists as a dimer. So this is a homodimer of two chains of the regulatory subunit. You have at the N terminus the dimerization domain that I just told you about. We have the cyclic AMP binding domains at the C terminus and this very disordered linker region. And so when we look at this linker region, you can see, first of all, this is all disordered in the free R subunit. And you have the inhibitor peptide that binds to the active site cleft. You have other motifs. There are phosphorylation sites. There's a putative GRAB2 binding site. There are many kinds of things that can be going on in this disordered region. So what happens when you bind and make a holoenzyme? Well, we've only seen a heterodimer, monomeric R with C, but we know under those conditions that this part becomes ordered. So that becomes ordered. So we're left with a small region that still seems to be quite disordered. So how do we get a handle on that? And so one way that you can do this is to actually introduce, so this is the part that's left. You can introduce cysteine residues. So do cysteine scanning mutagenesis and put fluorescent probes in and just look very generally at flexibility. How flexible is that residue when nothing is bound versus when you have a holoenzyme? And when we did this with David Johnson at Riverside, we found that these residues, all of these, in the absence of any ligand, cyclic AMP or C subunit, these were very, very flexible. When you added holoenzymes, some of them stayed flexible, but others became much less flexible. So this one at the end, for example, stayed flexible. This region here becomes very fixed. It's part of that tight holoenzyme complex here. But some of these became more fixed in the holoenzyme complex, and that led us to think maybe there's some other interactions there. So we made a new construct that actually extends further in this N-terminal direction. And we crystallized that to see, okay, is this communication going between the two linker regions that we might get some more ordering? And so Angela, this graduate student who carried out this work and got a crystal, but it was still a heterodimer. So it had in one asymmetric unit, a catalytic subunit and a monomeric regulatory subunit. But this tail did become ordered. And if we looked at that more closely, it didn't order all of it. There's still 10 residues or so missing, but it ordered about 10 more residues. And it ordered it in a way that it wasn't binding to its own cyclic nucleotide binding domain, and it wasn't binding to the catalytic subunit. And so we had to really go to the symmetry-related dimer to understand how this might be ordered. And so you can rotate it now. We're looking at it from the perspective, from the bottom of the dimerization domain, looking down on this. And so we can see that the N-terminal part of this inhibitor site is ordered by its interaction with the CMB domain of the symmetry-related dimer. And again, this is the inhibitor site for this dimer, and the N-terminus of that is coming over and interacting with this CMB domain. And so this provides an incredible opportunity for allostery, where we know the activation, the Hill coefficient for activating the heterodimer is about 1.2, whereas the Hill coefficient for activating this Tetrameric holoenzyme is 2.4, 2.3, 2.4. So there's an incredible amount of allostery that you achieve by having the tetrameric holoenzyme. And one can begin to see how you might achieve that here. You have the inhibitor peptide docking to both of the symmetry-related dimers, and then cyclic AMP binding. When it binds to this domain, it's going to unleash that catalytic or that inhibitory peptide from its own bound catalytic subunit. But you can see how it also has the potential to have an allosteric effect on releasing the inhibitor peptide that's bound to the other heterodimer. So this is very, very exciting for us. And although we we obviously need to do the structure of the full-length holoenzyme and validate this. This structure is of the tetrameric holoenzyme. The model that we create from this is very consistent with much information that we have about the tetrameric holoenzyme. So you can see here, as you rotate around, you pick up now a symmetry-related dimer, which gives you the framework for the tetrameric holoenzyme. As you go around, the next time you pick up the B domain, which is highly cooperative for this R1 alpha subunit. You must bind cyclic AMP first to the B domain, then to the A domain, and then you release the inhibitor peptide from the active site of the catalytic subunit. And then it's also a really nice model because the N-terminal part here is positioned so that it can beautifully link up to the dimerization domain. We have only about 20 residues that are missing from this structure now. So it's a very good model for really beginning to think about a tetrameric holoenzyme. And I think as we move forward, one of the real challenges for structural biologists who are working on these signaling proteins is to try to elucidate larger complexes. And I think from that we will really gain a much better understanding of how this works as an integrated system. And just an example of the R1 subunit, it has a class 2 PDZ motif at the C terminus, which is quite exposed here, and we're pretty sure there's some PDZ domain protein that's going to be binding there as well. So building this higher assembly of molecules is going to be a really important thing for all of us to try to achieve.